My name is John Stone. I work at University of Illinois. I'm a computer scientist and I work on developing a molecular visualization tool called VMD. VMD stands for Visual Molecular Dynamics. VMD is used for visualization of molecular dynamics simulations primarily, but it is also used for a variety of cell visualization and modeling tasks. How many people in here do some sort of computational biology, chemistry of some sort or another? Raise your hands. Okay, there's a few of you. I guess I saw out on the sheets out there, we also have a lot of people that are in math and computer science that don't really have a particular application domain. I can tell you there are all kinds of cool things going on in life science and computational biology, so maybe you'll get something out of my talk anyway, some of those people. So VMD is a little different than ParaView and Visit in that it is really sort of a bespoke tool organized around the kinds of workflows that are prevalent in these biological simulations at atomic detail. And it works with interchanging data coming originally from experimental imaging techniques of various kinds and bringing that data in, using that data to construct atomic detail models, which are then simulated on large supercomputers, and then we can visualize and analyze the results of those simulations. For any of you who want to try the VMD tutorials, while we don't have a hands-on session during the schedule today, later tonight or later in the week, you can try it at your own leisure. We have a large number of tutorials for VMD. Of course, besides these, there's also the VMD User's Guide. There's probably about 600, 700 pages of tutorials and material that you can read if you want. For those of you who are following along with my slides, the deck that I gave Marta actually has about 4 times the number of slides of what I'm presenting in the time slot today. So don't panic, we're not going to go through all that material. That's just there for you to read later or to have more details about the topics we discuss. I left section heading slides in so you can kind of keep track of where I am in the presentation versus the full presentation deck, just for your own benefit. So the role of VMD and the molecular simulation programs it interoperates with is to really provide the user with what we would think of as sort of a computational microscope. It allows scientists to study spatiotemporal scales that are beyond what any existing imaging modality is able to provide by itself. So we can use, you could say it's complementary to experimental imaging because we use the computer simulations to study the details that underlie some of the same things that an experimentalist might measure in a wet lab. So VMD interoperates with a lot of different software packages. Because it works with all of the different data file formats, you can use it in all the different stages of preparing, simulating, and analyzing one of these structures. It goes all the way from very tiny quantum chemical simulations all the way up to large cell simulations that are a scale of billions of particles. Many of the things that we're doing now in developing VMD are organized around enhancing its capabilities with what we call hybrid modeling. So these are techniques that combine classical mechanics simulations with either quantum chemistry or with coarse grain modeling techniques of various kinds. So we have different multimodal simulation approaches that are now becoming popular. And as you can see, it works with NAMD, AMBER, GROMACS, a long list of other programs. So those are all the prevalent tools that VMD works with. VMD is designed to be user extensible. So it isn't just a bunch of hard-coded C++ that is something that you can't modify or change. The code is open, but beyond that, it has built-in Tcl and Python scripting engines which allow you to write new software tools to automate complicated simulation protocols, to manipulate the data in various ways, to exchange different files with different tools. And so just to give you an example, one of the new tools that's in the latest versions of VMD is called QuickMD, and it's basically sort of you could think of it as like a simulation wizard. It guides researchers in their efforts to prepare, simulate, and analyze molecular dynamics simulations. It knows how to do hybrid QMMM simulations. And another thing it does is it keeps a log of all the different steps that are used to prepare the atomic structures and do all the different checks of those structures to make sure that they are viable for simulation. So this tool is interactive and is a graphical tool. We have hundreds of other tools built into VMD. Just for example, the ones that are green were ones written outside of our laboratory, so that you don't have to work closely with us to write extensions to VMD. Anybody can do this. And then these are just tools that have GUIs, like I have down in the bottom. There are about 75 other plugins which teach VMD to read and write various molecular structure file formats, and the list goes on and on. Some of the things that we've added in the most recent versions of VMD are support for Python 3. This is something that's very recent. We have new structure preparation and editing GUIs in the program. So there's a new tool called Multifacture, which it's been in VMD for a while, but it's been tremendously revised. And in the new version of the tool, it's a much more powerful part of the program. Some of the other things we've been working on are exploiting the performance that's available from multi-core CPUs and from massively parallel GPUs. We've used those to massively accelerate things like data clustering. So VMD has the ability to do high-performance structure clustering analysis built in. Another thing that we've exploited that with is for processing of volumetric data of various kinds, particularly tasks that are strongly correlated with experimental imaging data. So things like image segmentation, density map masking of various kinds, taking an atomic structure and creating a synthetic density map so you can compare an atomic structure versus an experimental structure. And then many other new and contributed plugins. A major area of technology development in VMD recently has been in ray tracing, and I'll tell you all about that coming up in later slides. And in adding support for remote visualization, because the data sizes that people work with now on state-of-the-art supercomputers have gotten so large, it's no longer practical to transfer that data off of those supercomputers. More of what we expect to do in the next several years is going to be done by remote visualization, either to a web browser or to a VMD client. So I'll tell you about some of those things. So VMD's approach to visualization is to produce a superposition of a bunch of graphical representation. So a graphical representation is a combination of a drawing style, a coloring style, and a grouping of other parameters that tell the program how to represent some part of the atomic structure. Representations also incorporate an atom selection, as we call it, and the atom selection understands the chemical structure that you're dealing with. So this is one of the things that makes VMD unique versus sort of general purpose tools. It actually understands molecular structures at a deep level. It has very high performance algorithms for walking the molecular graph, for doing various analytical tasks that involve understanding bond topology, things like this. Representations can be built up independent of each other, so you can make a visualization that draws, let's say, a coarse representation of the boundary surface of a molecule using, say, a molecular surface representation while showing the interior either in ball and stick or a so-called cartoon ribbon representation, things like this. And another thing that's cool about the way VMD uses its selection interface is that interface is shared between both the analytical parts of the program, the text mode interface, and the graphics and visualization features of the program. So the selection language in VMD operates a lot like the way you would use Google. So the way you would tell VMD to draw water within a certain range of the structure is with a selection like water within 10 of protein and Z greater than 0. So that's a simple atom selection. You can also use properties of the different amino acid residues. So you can use segment name BR. You can use the atom name or species, all sorts of other properties. And you can create user-defined fields for each of the atoms. They can be per atom or per residue or per segment. And you can fill those with any numeric data you want and use those in atom selections also. So another benefit of this kind of approach is these selections can be updated on the fly. So if you're doing let's say a trajectory animation and you have some time-varying structure, it will reevaluate those selections on every single time step. And so you can use that to your advantage. So this is a simple example showing a molecular surface representation for the water box combined with the ball stick model in the interior, and different shading and coloring modes being selected. So there are lots of different representations, and through the talk you'll see many, many such examples. VMD can compute various atomic structure properties internally. It understands things like ring structures. It can calculate things like pucker for various types of carbohydrate structures, for example. You can use those properties to color things. You can use them to draw those structures in different ways. And so all those things can be exploited to make informative images of various kinds. Other examples would be, so this is an example that's not built-in representation, but it's trivial to do. It's something you can do in about 3 lines of script, which is to calculate the per residue solvent accessible surface area. You could either do that for one time step, or you could do it for all time steps, average them together, and then use that to color the atomic structure, either in a surface representation like we've shown here, or with a ball stick model of various kinds. Assuming you have a structure that has all the charges defined on all the atoms, VMD can also do things like calculate electrostatic fields. It can either do a coarse grain calculation using particle mesh Ewald, or it can do Coulomb summation and get very high resolution electrostatic field plots. And so those are all things that are built into the program. It can do those both for a single time step and also for time averaging over the course of a trajectory. And using plugins like I was describing earlier, you can invoke things like principal component analysis on the structure and the trajectory to show the principal motions of different parts of the structure. So this is one that draws a so-called porcupine plot, so you can see where the primary motions are for those parts of the structure. VMD can represent things with a lot of different so-called material properties and coloring schemes. They can vary everything from sort of a normal looking molecular visualization to something that's more cartoonish to something that's more photorealistic. And I'll talk a little about the last case later on in the talk. So the key thing that differentiates VMD from other molecular visualization tools is that it was always designed for trajectory data. So that's maybe the thing that makes it the most different from tools like PyMol or Chimera or others. This is fundamental to everything that VMD does. That's why it works with the selection language, all the visualization modalities. This is just how VMD was from the very outset. So everything it does can be computed per time step. And not only can we draw those things for atomic structure, but you can extract information from the atomic structure also. So here's an example where we've taken a trajectory showing the ions vibrating around in the trajectory, and we have computed a 3D density map, probability densities showing where those ions like to spend their time. And then we've got a series of nested probability density isosurfaces, so you can see that, and then the trajectory is playing over the top of that. So this is an example of something you can do where you're going to produce a density map from something that happens over the course of the trajectory. So you can do that with occupancy, you can do that with atomic density and various other quantities. So one of the challenges we have working on state-of-the-art simulations is the growth in the size of the systems that people are now simulating. You can see this has gone up by orders of magnitude over the last 15, 20 years. It used to be the case that we would have 10,000 atom proteins, and now we're doing simulations of large photosynthetic organelles that are hundreds of millions of atoms, or proto-cells that I'll tell you about a little later that are billions of atoms. And so the problem there is, this is, it's historically the case that people would run their simulations on the supercomputer and they would download them to their desktop or laptop computer, and they would then do the bulk of their analysis on their local machines. But as the simulations have gotten larger and the time scale that's accessible to the state-of-the-art supercomputers has gotten longer, those trajectories have grown to the point that we now produce terabytes of output per day. And it's, I would say, commonplace for a state-of-the-art simulation to run for weeks or months at a time on one of these national centers. And so this means that in practice, unless a scientist wants to become an amateur rsync god, you would much rather do all of your work on the big machine. So we're putting a lot of our energy going forward into making it possible to do this, leaving the data where it was produced, and doing all the heavy lifting on the big iron, much like you've already heard from my colleagues this morning. So this is new for our field, though. But to give you an example, on the new Summit machine at Oak Ridge. This machine is so fast, running on about 20% of the machine. So we did this last year when they had only partially completed the machine. Running on only 20% of the machine, regardless of the system size, whether it was a 21 million atom system or a 200 million atom system, we produce about 10 terabytes of data per day in either of those system sizes due to the roughly linear scaling that NAMD achieves. So that's an example of where these things are going. So the reason why 20%, well, that's the amount of the machine that Oak Ridge wants you to be able to use for you to be able to run on that machine. If you can't run on 20% of the machine, then you won't be running on it. And so that guarantees that we're going to be producing data of at least that size. And that's being relatively conservative with the time steps that we save. So going forward, these next generation exascale machines offer tremendous opportunities in computational biology. Maybe the upper bound of where these things could go in the next 10 years is to simulate fractional cells. And so we just started a project to basically develop the modeling tools to facilitate this in VMD and in our other software. And we've been running what you would think of as a simulation of a cellular envelope on Summit. And this, just this shell, which has no cytoplasm or anything else, it's just the minimum amount of a cell that you can have to have sort of a viable model. Just that shell is 1 billion atoms, and just the structure, just the model of that atomic structure with the waters and solvent and ions is about 63 gigabytes. So you can imagine that it goes up from there as soon as we have dynamics. One time step is 12 gigabytes. And so if we store one time step every 10 picoseconds, we're going to have a huge amount of data coming out of a simulation like this. Now, this is a very early prototypical simulation. This is not yet a science project, you might say. But this is where we expect things could go with the exascale machines that will be built in the next several years. So this is very exciting. But there are all kinds of interesting practical challenges to visualizing and analyzing things on this scale. Nevertheless, even building the models. Building the model of the cytoplasm, for example, involves lots of structures that we don't have atomic detail structures for. So people are going to have to do that by analogy or homology models of various kinds. Fortunately, I'm hoping that next generation machines persist with a pattern similar to what we have with Summit. The details of Summit aren't that important in this context, but what is important is that the nodes on Summit are very powerful. So one node has about 0.5 terabyte of memory. It has a lot of processing horsepower. And so that means even one node of Summit can load an entire Proto-Cell structure without too much difficulty. So this means it's very aptly suited to doing the visualization and analytical tasks that we have in mind for a system of that size. And that's on a machine that exists now. Hopefully the machines that we have in a few years will be even more powerful than that. Let's see. And so some of the things that VMD supports that facilitate this are although it operates as a desktop application and you guys can download it and install it on your laptops this afternoon, when you run it on a supercomputer, it maintains the ability to do OpenGL rasterization using off-screen rendering, using what's called EGL. And it also gains the ability to do things like ray tracing and other high-end rendering techniques. So I'll give you a very short intro to what you can do running VMD on the supercomputer with MPI. If you want to try this, you have to use a build of VMD that has been compiled for MPI. The ones that we make available on our website are meant for normal desktop use. And so we have about 100,000 of those users. The number of people running it on the supercomputers today is relatively small. So what we've been doing up to this point is working with each of the supercomputer centers to get it installed with MPI support on their machine. But basically what VMD does when it's run on a supercomputer, you launch, the way I would normally recommend it is to run 1 MPI rank per node. That gives VMD the maximum amount of hardware per instance, so the largest amount of memory, the largest number of CPU cores, GPUs, etc. Within one node, VMD is heavily multi-threaded. It understands how to manage all the GPUs and CPU cores, and it will automatically figure out what's there and use it to do all the computing and visualization tasks that you ask it to do. So that's sort of something that you don't need to worry about as a user. Where the user gets involved is if you start running a large calculation on the supercomputer. Let's say you have 100 terabytes of trajectory data that you want to analyze. In a conventional serial program, you might write a for loop to process all the trajectory frames. Well, using the parallel version of VMD on the supercomputer, you can do that with we have a series of little script commands that begin with the keyword parallel, and then they do various things. So you can, of course, do all the sort of trivial MPI-like things like get your rank and the number of nodes and those things. But you can also do all reduce, all gather, and you can do a parallel for. You might ask, why don't I have complete MPI equivalency? And one of the reasons is we don't want users to, it's like handing them a hand grenade with the pin pulled. I don't want them to write scripts that create deadlock accidentally. So the APIs that we've exposed, VMD handles all the heavy lifting for you. So if you want to write your own script that has to do an all reduce or some sort of a gather from all the nodes, these are very easy to get right. It's hard to get these things wrong. Similarly, the parallel for implementation in VMD, it automatically schedules a list of work, and you don't have to do any load balancing. It has a built-in load balancer, so if you have a workload that is highly irregular, this built-in load balancer will take care of this for you. And so it'll automatically feed the work to the least busy nodes and all that stuff. So this is built into VMD, and it applies both to the CPU and the GPU algorithms, so you don't have to worry about any of those details. It works just as well either way. So just as an example, we ran a simple parallel analysis script on a Cray XK7, so this was on Blue Waters at University of Illinois. In a serial run on one node using a GPU would have taken about 14 days. We didn't actually do it, but that's what we guess it would have taken. Running on 128 nodes, it takes about 3 hours, and on 2,000 nodes, it takes about 19 minutes. And so this is to process a trajectory of frames comparing an atomic structure versus a cryo-electron density map and basically calculating the cross-correlation of all the different parts of the structure against the density map. So if we had done that without the GPU, that would have been something like 5 years in a serial piece of CPU code. So that's what we're comparing, that's the worst case scenario. But even with the single node version using all the CPUs and the GPU, that would have taken 2 weeks. So this is where, with a very small amount of scripting, you can take anything that you would have done in a conventional VMD analysis and spread it out over the entire machine. Another example of this, it doesn't just apply to supercomputers. You can do this on Amazon. So we have VMD running on the Amazon EC2 cloud. Just like you can do it on the supercomputer, you can do these parallel analyses or visualizations on the cloud. In this case, we did parallel movie rendering. So the workload of making a visualization, as you've already heard some of it today from our colleagues, the workload of producing those visualizations, it's not just the graphics. In fact, the graphics is actually, in some sense, the easy part, particularly with hardware like GPUs. Making the image is the last 3% of the work. The other part of the work is doing all the geometric calculations, producing meshes, maybe doing a bunch of physics calculations to calculate quantities that go into coloring the surfaces and so on. And so rendering a science movie may be much more computationally demanding than just making a rotating constant structure. If you're doing time-varying atom selections and you're deciding what to draw on a per time step basis, the math, there's more and more work. So if you parallelize over the frames in the movie, one of the things that's cool about this is with a small amount of scripting, you are able to parallelize all of that work. So not just the image rendering. We're getting fine-grained parallelism out of the multi-core CPUs and GPUs, and we're getting sort of weak scaling out of the analysis scripts or the visualization scripts that you write as a user. And so this is something you can do that's built into the program. It's very easy to use. So one of the recent advances in VMD is the use of ray tracing. So ray tracing as a computer graphics technique has been around as long as I have. And Its primary benefit is the ease with which it can incorporate advanced shading and lighting effects. So it can also directly render curved geometric surfaces, which for molecules, as you can imagine, is a big deal. We can directly represent spheres and cylinders and so on. And not only do they look better as a result of being directly rendered from their curved geometry, they're also more memory efficient. We don't have to draw a sphere as 1,000 triangles. We draw it as a sphere. And so we save a lot of memory by doing that. Other things that ray tracing is well suited to are stereoscopic rendering panoramas, and things like planetarium dome renderings. Anything that is a non-planar screen is something that conventional rasterization is not very well suited for. So if you want to do omnidirectional VR projection, like for let's say an Oculus Go head-mounted display, this is something we can do built in VMD as of the last version, in fact. And this is, not only can we do it in a batch mode rendering, but we can do it in real time. And so this is the result of a bunch of work that's happened over a long period of time. VMD has 3 different ray tracing engines built into it. The first one is Tachyon, which runs on all the different hardware platforms. That's one that I wrote. It also has an optics-based engine, which is a derivative of the first, which I wrote. That it's basically a subset of the first one, but it's very fast, and it uses the GPU and the latest hardware to do very high-performance interactive ray tracing. The last one is similar. It's a derivative of the first that's based on Intel Osprey. And likewise, it exploits the SIMD hardware instructions on the latest Intel CPUs to let you do interactive ray tracing on very large, complex scenes. So you might say, well, what do I want to do with that? Well, for our field, for those of you that are non-molecular scientists, you might say, well, why do these people care about this ray tracing stuff? The reason is we work with complex structures that have very corrugated, knotted surfaces that have pores, pockets, and cavities that are critical to their behavior, right? And we want to be able to see those things very easily without having to spend a lot of time. And so the benefit of a technique like ray tracing is we can implement advanced lighting models like ambient occlusion lighting very easily and very conveniently. And using a technique like ambient occlusion lighting, which is basically, if you imagine standing outside on a cloudy day where light comes from all directions, that's what ambient occlusion lighting is sort of emulating. And it brings out immediately all the pores, pockets, and cavities. These are the same structure. This is a virus structure that's been cut open, so you're seeing a half sphere of the virus. And on the left, you really can't see any detail at all, and on the right, it's obvious. And the thing that's beneficial about using ray tracing is with an advanced lighting model like ambient occlusion, the scientist doesn't have to spend any of their time positioning lights or becoming an expert in computer graphics. They get this result immediately. And so that's the idea. So again, for our kinds of geometry, ray tracing is beneficial because we can directly render curved surfaces. That saves us a lot of memory and gives us much higher quality images by default. And that makes it easier for us to bring out salient features like in this HIV capsid. You can see this little red circular region. Because we have ambient occlusion lighting, you can see where the pores in those hexamers are immediately. You don't have to highlight it. You don't have to label it or manually color it in some way to see it. You see it by virtue of the fact that the light casts shadows there. And so then that makes that kind of visualization a lot easier to produce. And to somebody who is familiar with these structures, you can immediately see that there's a pore there. And then this may be of interest because in this case, the pores allow ions to transport into and out of the capsid, and this is important for how it works. So some other examples are, here's a satellite tobacco mosaic virus. It sounds very evil, but it attacks tomatoes. It's not going to get you. So like everybody loves that title. They're like, wow, what is that? It's a virus that can't infect by itself. It's a satellite virus, so it can only infect something that's already got an infection by another virus. But these are another example of using those techniques. You can also use more photographically oriented techniques like depth of field to bring the eye to look at a particular part of the structure in great detail, or to provide the entire structure for context, but only show a region of the structure in sharp focus. As I've said, we've been working on the ray tracing for a number of years. On the high end, the challenge is rendering huge structures with hundreds of millions of objects. So this is a photosynthetic organelle called a chromatophore, and it has about 100 million objects in it just in the little piece that you see with all the lipids in the membrane. And to draw that interactively with OpenGL or other techniques is very difficult because of all the spheres and all the other glyph geometry that has to be drawn. This is an example of something that's perfectly well suited for ray tracing techniques. Again, we can do omnidirectional renderings for VR. I'll probably show you a little tiny piece of one of those. You may have heard a lot of the news recently, particularly if your hobbies involve any video games. NVIDIA has had a new generation of GPUs, and they have a hardware feature called RTX that accelerates ray tracing specifically. And you might ask, well, what good does that do for scientific visualization? In our application, running the same light harvesting organelle structure, the difference is with this kind of hardware acceleration, we're able to render the same picture that we did before about 8 times faster now. And so for us, that's a big deal. That's comparing the mid-range state-of-the-art GPU versus what had been one of the highest-end GPUs that they sold previously. We're expecting that Intel and AMD will also have hardware-accelerated ray tracing, so I think in the next several years we're going to see a tremendous uptick in the use of these advanced rendering methods. And these are how we often render high-quality images for journal covers and things like this, and just because they're easier to interpret. I mentioned earlier that because ray tracing is able to do non-planar projections, we can do things like dome planetariums. One of the things we've done in the last year was we were part of a dome show called The Birth of Planet Earth, which we show some of those photosynthetic organelles as they've evolved and how they basically gather sunlight and turn that into chemical energy. And that show involved this kind of panoramic rendering. And so another example of which is VR HMDs. So how many of you have ever heard of the YouTube 360 videos? How many have ever seen one of those before? Okay, so about half. So YouTube, aside from having the normal cat videos, has now a feature for doing omnidirectional projections. So that means you have a movie that you can actually turn around in a complete circle and see in all directions. And not only that, but it's in stereo, so it's 3D. It feels like you're actually there. So VMD has the ability to render these omnidirectional projections using ray tracing, and you can render a complete movie using the built-in tools in VMD, and you can upload it to YouTube. You can also play it in an Oculus Go or any of the other state-of-the-art HMDs. And this is great, particularly if you're taking a movie to something like a poster session. You can basically take an Oculus Go, stand next to your poster, and if somebody wants to see your movie in 3D, it's not like the old days where they'd have to cross their eyes or something on a printed page. I mean, those nightmares are over. I can't even do that, to be honest. I am unable to do it with eye crossing. I just give up. But so now they can see an animated movie. It's 3-dimensional and it's omnidirectional. So you can also fly the camera through the scene and do all the usual cinematic things that you would want to do. But this gives the user the freedom to look around in a way that they didn't have before. And so this is really exciting. One thing I wanted to mention about this, going beyond this, I mentioned that we are working heavily on remote visualization. One of the things we want to enable is the use of head-mounted displays from a distant supercomputer. So this is a difficult problem. This is basically the same thing as the YouTube omnidirectional 360 movies, except that we are streaming the video in real time from the supercomputer to a headset. So in a similar way, you'll be able to look around in all directions, but you can also navigate and you can change the representations and interact with the scene, which is something you can't do in a canned movie. And so this is something that's in development now. We have lots of prototypes implemented already. And what's exciting about the speed benefit that I mentioned from the state-of-the-art hardware is that factor of 8 performance gain means this can be done with a conventional desktop machine now. To do it before, I needed something like 16 GPUs. Now, one state-of-the-art GPU can do the same thing today that used to, in 2016, take 16 GPUs. So that's really exciting. So some of this that I've described, for those of you that are computer scientists who are interested in visualization and rendering, there's a book out. It's freely available. You can download it as a PDF, and it's got a huge number of great articles on ray tracing technology. And I wrote 2 chapters in there, one on the planetarium projections and the other on using ray tracing for scientific visualization. So you can go check that book out. So in the last couple minutes, I'll just summarize some of the other cool things that we've been doing. We've been accelerating density map segmentation. One of the challenges that you have when you have an experimental image that contains a noisy density map, unlike atomic structures where we have, it's very easy for us to write code that selects parts of the chemical structure according to their attributes. When your only attribute is some density value, it's difficult to disambiguate one part of the structure from the other. So there are algorithms known as segmentation algorithms that will let you use the computer to find connected parts of the density map geometry and label them. And once they've been labeled, now I have a way of reasoning about that part of the structure. So then I can apply different coloring to that part of the structure. I can use it as a selection. I can calculate various properties. And so we have a very fast image segmentation tool that works on these volumes. Going forward, we're applying the same thing to larger data, such as tomograms, and we want to be able to do things like fitting different high-resolution cryo-EM data to lower-resolution tomograms using some of these techniques. The math behind some of them is very closely related. We have cross-correlation algorithms on the GPU also that are very fast. And so to give you an example, we've used those cross-correlation algorithms to evaluate quality of fit for atomic structures against experimental density maps. And using the GPU, this is now extremely fast compared to conventional software, so we can do much more computationally demanding calculations than were previously feasible. So these are some examples running on machines that are similar to the node architecture of Summit. So, comparing to normal software, this is in the hundreds of times faster. Compared to our own code, the latest versions are now about 10 times faster than what we did just about 3 years ago. So that's very exciting. And one of the other things that we're putting a lot of energy into is clustering of molecular dynamics trajectories. So this is used to elucidate different transition points over the course of a structure. We do an all pairs n-squared comparison of every structure in the trajectory versus every other structure. So this produces this little upper triangular matrix of root mean square displacements. That goes into a clustering algorithm, and then out pop, well, not quite this simple, but out pop a number of clusters that are then families of related structures. And our goal here is to make this extremely fast, much faster. You can do this in tools like R and MATLAB and other available software, but by writing our own GPU code for this, we are in the range of hundreds of times faster than any of those tools. And of course, ours are sort of cultivated specifically for molecular dynamics trajectories. So this gives us a tremendous advantage. Some of the exciting things that are maybe coming in the next several years that are technological are non-volatile DIMMs. So how many here have ever heard of persistent memory? Okay, so did you guys already hear it this last 2 weeks? Okay, so you're well informed. I'm impressed. Most people don't know what persistent memory is. So one of the opportunities that persistent memory represents is the opportunity for us to work with very large structures like these cell scale models. If you're working on something like that day in and day out or frequently, the size of the structure data that you're working with is huge. And if you read all the input files to reproduce the state every time you load the software, you can imagine this is very time consuming. So one of the opportunities we have is to basically have persistent in-memory data structures, not like a file, but rather like pointers and C memory buffers and C++ objects will reside in persistent memory. And this would allow a program like VMD to essentially load and in a matter of a quarter of a second, reconnect a bunch of pointers and data structures and work on something that might be a terabyte or 2 terabyte data set interactively. And so this is something that's never been possible before. If I have to read poorly structured file formats like PDB files or other similarly low density sources of information, doing the same thing might take 6 or 7 hours rather than being instantaneous. This is going to be a game changer for people that want to do comparison of one structure or one family of biological organism or protein versus other proteins. You could have the entire protein data bank loaded in the program all the time. And so that's kind of an interesting opportunity. You should think, and I would love to hear ideas from people in the field. If you had the ability to load a few terabytes or have it available with an already loaded set of structures, sequences, or other data, what would you want to do with that? So this is something, we'll see first on high-end HPC machines, but I expect eventually this will make its way into things like laptops, and that'll be a huge opportunity coming our way. And so with that, I'll take any of your questions. You have many. Well, let's start. Let's try the first one. Ask the question that you think they're most interested in.
How do I, starting from my, could you go through we have VMD, could you go through an example and start from, just plot like a PDB and then get it to look very cool.
You want the journal article, the least resistance path to the journal cover? We probably, so first we should probably sit for that. And we should get you, see what you've got for a laptop there. But, so I guess it depends, the path, least path of resistance to a journal cover hinges upon the size of the system you're viewing. So that has a lot to do with what visualization modality would be prevalent. Like, would you show it as a ribbon structure? Do you want to, what is the salient detail? What's the important part about the structure you want to convey? Is it secondary structure? Is it the configuration, atomic detail configuration or close proximity of surfaces, surface interactions? Is it some other feature? You have to, you should make a list of what is it that you're trying to communicate by making that image. Because, it isn't about a pretty picture as much as a communicative picture. And so you should make a list of when somebody sees this image, I want them to see this, and I want them to see that, and I want them to see this, and I want it to imply this. And so you're telling a story even though it is a fixed image. And with molecular dynamics, this is half of the challenge. Part of the reason we have all these fancy rendering techniques is if you are making a journal figure, you don't have the opportunity to show that dynamics interactively. So you're trying to make a still image that communicates as much as possible. And so, but I would begin by making that list. And then there are sort of techniques for each one of those things that you can use to create that effect or to show that detail in a way that's easy for somebody else to see, particularly somebody who's trained in interpreting those images. But you can come over and we can sit. All right. All right. Yeah?
So my group's interested in adapting the optics geometry library, but use that with our physics.
Sure. When you say geometry library.
So use the ray tracing. So we're transporting photons, but
sure.
they're X-rays.
Yeah.
But they behave essentially the same as visual light, right?
Yeah, I've used it for RF propagation too. So you can do quite a few things with ray tracing, not just graphics.
Right. So I'm curious. So you used it for RF propagation. Did you have any issues with the precision limitations?
No, we didn't. But we had, so I wrote software for a company called Delcross that then got bought by Ansys. And I guess I have to be careful what I say. So their approach involves multiple precisions. And you can use one precision as screening, excluding candidates and things like this. And you can do, for something that's a hit or something that's accepted, you could do a double precision calculation. So in optics, the way that rays are traversed through the bounding volume hierarchy is all done in single precision. So deciding what box that the ray hits is done in single precision. If you need a ray object intersection that's double precision, one of the beautiful things about optics is you can do that easily. And if you're worried about the precision of the ray box intersection, you can expand the box slightly to be slightly conservative, and then you'll be guaranteed to get a hit on the box that contains the object, the bounding box. And then all you have to do is write the double precision ray intersection yourself, and you can basically use that as per ray payload. So you could record your double precision intersection along with the single precision intersection t value.
So when it hits the bounding box, then it switches over to the double precision?
Well, it'll run your intersection test. So that's the trick, is to run your intersection test rather than a canned one. Yeah.
Because, yeah, one of the concerns is, I mean, if you have like a bunch of tets, right,
Mhm.
you can have many vertices join, and then, you know, if you don't have high enough precision at that intersection, then
That's
you
right.
don't know where you're at. So.
That's true. And yeah, I think there I would think about the plane equations for those tetrahedra faces. There's a bunch. One of the things in the Ray Tracing Gems book is a very, very detailed discussion about numerical precision issues, where they show all kinds, the way you write the ray intersection code, which order you evaluate certain operands will change the precision due to the magnitude of the floating point numbers, because floating point is non-associative, and so anyway, there's some details to get right there, but there are actually quite a few published algorithms for getting high precision on different types of geometries, and if you have any questions, I know who to send you to ask for the oracle of such things, so yes.
And the Intel Osprey,
Uh-huh.
so we explored the Intel Embry
Yeah.
library. but
It's kind of the same deal with
okay.
optics.
Okay.
Optics, you could argue that they cover the same territory. They have some of the same issues. They are both of them are kind of focused on single precision workloads, but I think in both cases there are schemes by which you can exploit them for a higher precision work by using them as an accelerator and then following it up with a full precision math calculation of your own. Yeah, that's what I would advise anybody to do, is to use that approach.
Okay, thank you.
Other questions? Okay.